Hello everybody and welcome to the Mutagenesis Lab. Today we're going to be mutating some bacteria so that hopefully they'll be able to grow on sodium azide plates, which normally don't allow for bacterial growth. So the first step that we're going to do is to transfer a culture that we had already pre-made. We're going to transfer 0.1 milliliters from the culture into the test tube 1, which already has 9.9 milliliters of broth. And this is to dilute the bacteria. So here we transferred it and we're mixing it up. That just makes sure that all of the bacteria are equally distributed so that when we pipette out of it from test tube 1 into test tube 2, we're getting an even mixture. So again, we're transferring 0.1 milliliters of fluid from test tube 1 into test tube 2. Before and after you transfer, you want to make sure that you flame the test tube so that you aren't transferring any other bacteria to the tube. And again, we're going to mix it up so that there's an equal distribution. For step 3, we're going to take 1 milliliter, not 0.1, but 1 milliliter from tube 2 to tube 3. So this time we're going to use the actual pipetter. Mix it up. And then now we're going to take another 1 milliliter and transfer it from 3 to 4. And now that we have all of these test tubes, we have to pipette 0.1 milliliters from test tube 1 onto the sodium azide A and B. And make sure that you label the plate with your initials, what the plate is, the date, and the plate letter. And we're going to mix up test tube 1. Flame the tube to make sure that you don't get any bacteria in it. And then we're going to take 0.1 milliliters of the bacteria out and plate them onto plates A and B. So here we're transferring the 0.1 to each of the plates. Now that that's done, we have to do the other two plates. These ones just have the regular nutrient agar without any of the sodium azide. And again, we're going to take 0.1 milliliters and put it onto the plates. So test tube 3, we're going to take 0.1 milliliters and transfer it to plate C. And then we're going to transfer 0.1 milliliters from test tube 4 to plate D. Alright, and now that all of the bacteria is on their respective plates, we have to spread out the droplets so that they're equally distributed along the agar. So to do that, we have a glass spreading rod with 70% ethanol, and we're going to flame the ethanol so that it's off. We make sure to cool the glass spreader in a part of the agar that does not have the bacteria, and then spread the droplet out evenly across the agar. Put it back in the ethanol. Then we're going to flame it again and repeat the steps. The ethanol and the flaming are so that we make sure that the glass spreader is completely sterile so that we aren't transferring one group of bacteria to another plate. And so now that the first set, the pre-mutagenesis bacteria, has been made, we're going to take the rest of the bacteria from the culture and we are going to hopefully get some mutations by putting it under a UV light. So we take the remaining culture that we have in the test tube from the beginning and we're going to pour it out onto a glass petri dish. Then we're going to set a timer for 1 minute, move it into the UV, and then when we're ready, we remove the lid and restart the timer. After 1 minute, we're going to take it away from the UV light, and then we're going to transfer 1 milliliter from this dish into a small flask that has 9 milliliters of broth. And then after this, we're going to cover it in tinfoil. I want you guys to try to figure out why we're doing that step. And then once it's wrapped in tinfoil, we're going to incubate the flask at 37 degrees Celsius for 1 hour to let the bacteria grow. So here's the flowchart for the post-mutagenesis plates that we will be making, and we will go through this step by step. So the first thing that we want to do is we want to transfer 0.1 milliliters from the flask with the UV exposed bacteria to test tube 5. Next, we're going to mix that up to make sure that the bacteria is equally distributed within the liquid. Make sure to cover the flask back up so that you don't accidentally tip it over. And then on to step 2. We're going to transfer 1 milliliter from test tube 5 to test tube 6. All right, and after we have that, we're going to mix it up so that it's equally distributed again. And then we're going to transfer 1 milliliter from test tube 6 to test tube 7. Make sure to mix it up. All right, and now the test tubes are ready to be put onto the plates. So now we're going to take 0.1 milliliters from tube 5, and we are going to transfer it to plates E and F, which have the sodium azide. So for this, we took out 0.2 milliliters and we just transferred 0.1 to each plate. Because they're coming from the same test tube, we are able to do that. Next, we're going to transfer 0.1 milliliters from test tube 6 onto plate G, which does not have the sodium azide. Now that we're done with that, it's time for the last transfer. We're going to transfer 0.1 milliliters from test tube 7 onto plate H, which also does not have the sodium azide. All right, so now all of the plates are made. It is time to, once again, spread out the droplets so that it's equally distributed along the auger. So again, we're going to do the ethanol, flame, and then spread, making sure to cool off the glass spreader and a part of the auger that does not have the bacteria so that we don't kill the bacteria. When you do this, try to open the lid as little as possible. Here a couple times the lid is taken off completely, but that could include some contamination, so generally you want to keep the lid as close as possible while spreading it. Like this. All right, and last plate. And we are done creating the plates. So now we're going to let those sit and then put them in the incubator for 24 hours before counting the cultures. Here you can see that on plates A and B with the sodium azide, there is no bacterial growth, while on C and D, there is bacterial growth. This is what we would expect from the pre-mutagenesis ones. And now E, F, G, and H. E and F also do not have bacterial growth, while G and H do. It's harder for bacteria to grow on the sodium azide, even if they have the mutation. So now after 48 hours, we'll see that plates A and B still don't have any bacterial growth, while plates C and D do have bacterial growth. Now, why do you think that plate C has more than plate D? And then for plates E, F, G, and H, E and F now have bacterial growth, and G and H also have bacterial growth.